Jean Swidler. I'm with Genetic ALS and FTD and the Legacy, an organization dedicated to the needs and interests of the genetic ALS and FTD community. My family has the C9 repeat expansion. I confirmed I carry it pre-symptomatically or asymptomatically. And I can't wait to hear from our guest presenter today, Dr. Andrew Douglas. And with that, I'll let you take it away, Doctor.
Great, thanks very much, Jean. I'm going to try to share again here. So, okay, how's that? Is that coming up?
Yes, looks great.
Great. Okay, so I'll just kick off then. So thanks very much for inviting me to give a talk. I think this is essentially based on a paper that we've just published and which has, I think, the same title, essentially Personalised Penetrance Estimation for C9orf72 ALS and FTD. So that's what I'll talk about. And let's just move on here. There we go. So it's always hard to know exactly how to pitch it, but I thought I'm actually a clinical geneticist, or if you're in the US, I think you'd call it a medical geneticist. And so I thought I'd just start off talking about ALS and FTD and how it applies to clinical genetics, and then move on to talking about this idea of reduced disease penetrance with reference to ALS and FTD, and then think about actually what is the penetrance of genetic forms of ALS, and then talk about this actual estimation of the penetrance in individual families, and then a little bit at the end about, well, what is it we might be missing here, and why is it that this does seem to vary from person to person. Oh yes, and this is to remind me to introduce myself. So I am, as I said, a medical geneticist, a clinical geneticist in Oxford here in the UK, and I work primarily actually at the Nuffield Orthopaedic Centre, although I'm nothing to do with orthopaedics, but that's where the clinical genetics department is based. But I also do research with the MND research group in the John Radcliffe Hospital, which is headed up by Kevin Talbot. And on the right there, you see a picture of the Churchill Hospital from 1940, which is where our clinical genetics laboratory is based. It's hard to find an up-to-date picture that looks flattering, so I thought I'd show the 1940s picture. It was originally set up in the Second World War, actually under the US military for servicemen coming back from the war, but it still functions now as a hospital. So that's a bit about me. All right, so what about ALS and FTD in clinical genetics? So I'm assuming that this audience knows, so I'm not really going to introduce ALS and FTD specifically, but in the clinical genetics clinic, I'm usually faced with families, or family histories at least, that might look a bit like this. And hopefully you're familiar with how this is. We have essentially males and females. Males are squares and females are circles here. And depending on what you're looking at, if somebody's affected by a condition, you color them in to indicate that. And if they've passed away, you put a line through them. You may have some details about their ages and things like that. So sometimes we'll be referred people who are affected by ALS or FTD or other conditions, and they haven't had any sort of genetic testing. And if you had a family history like this, where there seems to be 3 generations affected by neurodegeneration with a mixture of ALS and dementia, then you would think, well, that's a bit suspicious that there could be a genetic cause. And so you might want to do a genetic test, and specifically a diagnostic genetic test to confirm that there's maybe a mutation traveling down in the family. And so the people coming in this scenario would usually be asking, you know, I've got a family history. Is there a genetic cause for this condition in my family? And then the next thing to ask is, well, actually, are my children at risk of getting this, or my other relatives? And increasingly now, people may want to know if they're eligible for any specific gene-based therapy or any trial. And so increasingly, we're not the ones in the genetics clinic doing this testing, but the neurologists themselves are doing this sort of testing straight off the bat, as it were. But the other, and perhaps the more common situation for us in clinical genetics is that we see unaffected relatives who may have a family history of ALS or FTD, and they want to have predictive genetic testing, or to at least to consider it. And their questions are likely to be, am I actually going to get this condition? And I want to know, because I may want to change something about how I'm living my life, or make decisions about having children. And indeed, a key question is, if I do have it, am I going to pass this on? And can I make sure this isn't going to happen? So this is sort of the other type of situation that we often see in the clinic. And it's perhaps more clear-cut when the family history is really obvious like that there's something. But very often, this is almost an identical family history, but it's not quite. And this can often be the case, that it's not often straightforward, that there may be information that's missing, or it may just be somebody hasn't been diagnosed yet. And so it can often look a bit less suspicious, just because it can vary from person to person, from family to family, whether someone gets a diagnosis. They may have another illness. That means they don't end up living long enough to get one of these conditions. And a couple of generations back, you simply may not have had any specific typing of your dementia, and actually they may still be the case today. So yes, often it's not quite so straightforward. And as I think you'll know, it's also the case that not everyone who inherits a gene mutation like that is going to get the disease. And part of that is because people may die from some other cause. And another reason is because there is this phenomenon of variable penetrance, which we'll come on to. Okay, so I thought I ought to say something about what sort of testing we do. So in the sort of diagnostic genetic testing sense, most commonly, this is changing, but at the moment, it's still more common for someone not to have had genetic testing in their family before. And so we don't actually know quite what we're looking for. So if somebody's affected and they have a family history, occasionally we will know what it is that we're looking for, but often we don't. And so we need to actually do a broad test looking at lots of different genes. And in the UK, at least, this is now done by a very broad test, which is actually whole genome sequencing. So all of your DNA is basically extracted, and it's cut up into little pieces like this. And then essentially all those little pieces are stuck onto a chip, basically, and read in parallel. So you get all these short segments of letters of genetic code. And then you sort of, like a jigsaw, sort of patch those back together to what we know should be the sort of normal reference sequence of the human genome. And as you do that, you will start to see little changes that are in the sequence of the individual that are not present in what's supposed to be the normal sequence. And what you do then is you've actually got all the genome data, but you just focus in on those genes that we know are linked to ALS, and look specifically if they have any changes in them, and any changes that are likely to affect how the gene works and basically be responsible for the condition. So that's quite a difficult, complex process. You're looking at a lot of information and a lot of data, and you have to be sure of what you're finding, because each one of us will have maybe 3 to 6 million changes from what's supposed to be the reference normal sequence. And so it's quite hard sometimes to be certain if a change you found is actually one that causes disease, or it's just part of normal human variation. So hopefully in the end we get an answer, but it's quite different in the situation of a predictive genetic test, because this is where we are testing an unaffected relative, and we have to already know what we're looking for. So their parent or sibling will have already had this sort of broader test and will have had a specific mutation change in a specific gene. And in that setting, actually, all you're going to do is look for that same specific gene change in the unaffected person. And that is a targeted test, so you don't have to do all this genome sequencing. You just have to look at the one little spelling change or the repeat expansion if it's C9orf72. And I've just put here an example of that upper little trace is what we call Sanger sequencing, and the little peaks are different letters in the DNA code. And you'll see in the middle there, one letter has changed from a C to a T, and that's all it is. In the bottom panel here, we've got the sort of tests you might do to look for C9orf72. It's a different sort of technique, but it's also it's something called repeat primed PCR. You try to basically amplify the bit of DNA that's expanded, and you end up with this little sawtooth pattern that shows that actually the repeat is present multiple times, and that indicates there's an expansion there. But as a targeted test, it doesn't tell you about all the other genes. It's very specific. All right, so having said I wasn't going to introduce ALS, I'll just say something about genetic forms of ALS. And I think you'll probably know, obviously, ALS is actually not as rare as all that. It's affecting 1 in 300 to 400 people, and it's present worldwide. And you often will see this figure that 5 to 10% of cases seem to be familial, although actually, if you look at it, it's not very consistent how that's defined, which I'll come on to later. But in general, people say 5 to 10% of cases are familial. And when it is familial, then we tend to be able to find a specific genetic cause, perhaps 60 to 70% of the time. So that does mean there's 30 to 40% where we still don't really know why it's running in the family. But for the majority of cases now, we can find an answer. And then there is this overlap with frontotemporal dementia, and nearly a quarter of people with ALS can get diagnosed with frontotemporal dementia, and a larger proportion would have other sort of changes that might be tending towards that, but might not get a formal diagnosis. And this is a picture that's a few years old now, but you'll see the years on the bottom there and the different genes that have been discovered up to now in order. And the size of the chart in the upper half, that's the proportion of familial cases of ALS, and in the bottom half, it's the proportion of non-familial, apparently sporadic cases that are explained by genetic causes. And you'll see that obviously the biggest step up there is around 2011 when the C9orf72 expansion was discovered. But then SOD1 was the first one, and the whole panoply of others have been discovered in the meantime. But interestingly, you look at the shape of the graph on the bottom, and I've always thought, well, it looks kind of similar. It's just smaller and more squashed. And so we do find in cases that are sporadic, that don't seem to have a family history, that actually we can sometimes find the same genes. And it really is the same genes, which has always been striking to me and is what has led on to us starting to study this penetrance. And the other side of the coin is the frontotemporal dementia, which often does have a familial component. It's really hard to actually find figures for how common frontotemporal dementia is. It is not the most common form of dementia, that's Alzheimer's disease, but actually how much of dementia it really causes is a bit unclear, and you can find lots of different pie charts that give you slightly different figures. But it's supposed to be the 4th most common dementia, and actually it's on a par with ALS in terms of how common it is, at least as far as we can see. And interestingly, it's not so different in that 10 to 15% of cases seem clearly familial, but actually a lot of more cases, maybe 40%, have some sort of family history involved. And there is also that overlap that maybe 10 to 15% of people with FTD can also get diagnosed with ALS. Now, the FTD community perhaps has been a little bit more involved in working out what they mean by familial, and they have this sort of Goldman score, which you can use to work out what is the chance of there being a genetic mutation in the family. And if you have a clearly what we call autosomal dominant inheritance, so it's lots of generations, and in fact, well, they say 2 generations with a first-degree link, and you need 3 relatives, then you've got a really high chance of finding a genetic mutation. Whereas if you only have perhaps one affected first-degree relative over the age of 65, it's only about 12% chance. And if you have nobody else in the family affected by FTD, then it's only 7% chance. So that gives you an idea of how likely you are to find a mutation in FTD. And on the right here, I've just put this figure from a paper that looked at what we say the clinical types of dementia. So Alzheimer's disease, frontotemporal dementia, dementia with motor problems, and then as a prion disease, the thing that gives you CJD, Creutzfeldt-Jakob disease, and then what genes were involved in those cases. And you'll see there's a little bit of overlap and crossover between these quite different clinical syndromes and actually the genes. And that's telling you it's a bit more complicated than we originally thought. It can be actually quite hard to tell what sort of dementia somebody has just from clinically examining them. And I think it complicates analysis of family history because dementia is so common, and I'll come back to that. So anyway, over the past 10, 15 years, it's become quite clear that there is this continuum of disease between motor neuron disease, ALS, and frontotemporal dementia. And different genes do seem to play a part. And some genes usually always give you ALS, and some genes usually always give you FTD, and some are in the middle. And C9orf72 expansion really is right in the middle, and it's probably similar, your chances of getting ALS and getting FTD, or both. So that's this sort of spectrum of disease. And as I've said already, you tend to find the same genes. There's a bit of crossover between FTD and ALS, but in terms of familial cases, you really do see the same genes just represented at lower levels in the people who don't have a family history. And so that's telling you that there's something going on there, that it isn't just that you will definitely get the disease if you have a faulty gene. And you can look across different parts of the world and you find the same thing. So here are some different parts of the world, and the inner circles here represent familial cases of ALS, and the outer circles in the same populations represent these sporadic cases. And the different colors here are the different genes. In Europe, North America, Australia, C9orf72 is the biggest part of the wedge of the circle. But you'll notice that actually, if you look at the outer circles of the sporadic cases, it, to be honest, just looks like they've been squashed together. It's a similar sort of pattern, just that there's fewer, a smaller size. So that's pretty consistent across different parts of the world, although the genes responsible do vary, and particularly in Asia, there's very little C9orf72 responsible. All right, so this kind of led on to the idea that there must be this reduced disease penetrance, and I ought to explain what that is. So I'm a geneticist, so when I'm thinking about penetrance, it's really the proportion of people who have a given genotype, that is a specific faulty gene, who actually go on to develop the related phenotype, that is the condition in question, ALS. And if you have incomplete or reduced penetrance, it means that it's not 100%. So not everybody with a given genotype, with a given faulty gene, develops the disease, the condition. And what are some examples of this? Well, perhaps a well-known example of a fully penetrant genetic condition is achondroplasia. So, people with this condition, they nearly always have exactly the same change in this gene called FGFR3. And it affects the cartilage of the ends of the bones, and the bones don't grow normally, and they always have the achondroplasia condition. They always have very short stature and distinctive sort of look to how they are. And so you will always get that condition if you have that mutation. That's one fully penetrant condition. However, here's an electrocardiogram of heartbeat, and you can get this condition called long QT syndrome. This is just saying that the length of time between when the heart ventricles contract and when they relax, basically, is too long. And if you have that condition, you are at a higher risk of having a funny heartbeat and actually, unfortunately, the heart stopping suddenly. However, if you inherit a faulty gene, this KCNQ1 gene that can cause this condition, you actually perhaps have a 65% chance of having this long QT syndrome. And so this is not a fully penetrant condition. This is an incompletely penetrant condition. So I hope that makes sense, but how do we apply this to genetic forms of ALS? We did a project to try and look at this, and because we're doing more and more testing of people's genomes and collecting lots more data in big databases, it's now possible to actually look at this in a sort of population-wide way, because we know how common ALS is, and we know how common it is to have a genetic mutation in each of the genes if you have ALS. And we also know how common it is for ALS to be familial. So we know all those things, and so we can work out in the population how often we ought to see specific mutations. So we can go and do that. If we have too many mutations compared to what we know about the epidemiology of the disease, then we can say that actually this disease is not fully penetrant. And so we did this looking at this database called NOMAD, which we use a lot in clinical genetics. It's a big aggregated database, I think, out of the Broad Institute in the US. There's hundreds of thousands of people who, from different studies and cohorts, many of them with adult onset diseases, heart diseases, respiratory diseases, things like that, but not so much ALS, although there are some who will have ALS. But it's used as a big repository of genomic information. And the other part of that is that it also includes information from the ClinVar database. Now, this is a sort of, it's an NIH-based repository of information about specific genetic variants and whether they're disease-causing or not. And so those two are actually integrated, and you can see which gene variants in NOMAD are called as what we call pathogenic, disease-causing in ClinVar. So that very easily and quickly gives you an idea of how many people in this database, which is some hundreds of thousands, have got disease-causing mutations in specific genes. So we can work this out, and we know the lifetime risk of ALS. We know familial cases of 10%. We know how many people get FTD. It works out at about 1 in 300 as well. We know we're going to say 1 in 10 cases of familial. And then we just look and see whether we have the expected numbers of cases in NOMAD. And don't worry about this, but essentially, for each gene, what we can say is that the expected frequency of the gene in the population ought to be the chance of you getting ALS, okay, maybe 1 in 300, multiplied by the proportion of familial cases explained by the gene, say C9orf72, and the proportion of sporadic cases of the condition caused by the same gene. And you multiply all those together, and you also do the same thing for frontotemporal dementia, because it can also be caused by, for example, C9orf72. And then you work out if that is what you've got. That's the expected gene carrier frequency. And the penetrance then is simply the expected gene carrier frequency over what you observe in the database or in control cohorts, just like that. And don't worry about this table. This is just showing you all the data and how many we looked at. We looked at the 4 most common genes linked to ALS. And I'll display it in a more straightforward way here, I think. So we've got C9orf72, SOD1, TARDBP, and FUS. And you've got these circles here. The orange circles are the kind of relative proportion of disease-causing variants you would expect. And the blue one, the bigger one, is the actual observed number. And so what that comes out as is that for C9orf72, we got a penetrance of 33% at a population level, whereas for SOD1 it's 54%, and TARDBP and FUS, again, under 40%. So we thought this was quite interesting and sort of tallies up with those pictures of those familial versus sporadic cases and the proportions of people who have these kind of gene mutations. And we published that in the Journal of Medical Genetics, and you can go and have a look at that. So what does this mean? What's the implication here? Well, it means that at least in the general population, it's not exactly the population, but it's a reasonable population database. The penetrance of genetic forms of ALS is reduced. and not just in C9orf72, but in other genes as well. However, we also have to remember that clearly there are families where it really seems to be very highly penetrant, and that the risk of an individual getting it is very high in some families. So these are two different things, and you can't just say, oh, therefore everybody who has inherited C9orf72 has got a 33% chance of getting it. That's not true. And so actually what you need to look at is the family history of the individual. So I mean, there must be some cause for this, and that's a bigger question that I'll come back to. But I thought we ought to be able to start to work out a personalized risk figure for an individual, because it's clearly not the same for everybody. And this is important because well, for one thing, people are starting to do clinical trials and thinking about even doing pre-symptomatic clinical trials. And actually, if you're somebody who you may carry a faulty gene, but you might be lucky that you are never going to get it, even if you live to old age. And if you were to then be put into a clinical trial, that might not be helpful. For one, you might need to undergo rather invasive intrathecal injections of a drug for the rest of your life, and you maybe never needed to do that. And also, might bias the results of that clinical trial, in that actually it's not going to show that the drug is helpful. So I think that's quite important. But the other thing, I suppose, is that knowing your own risk, if you can know it, it's helpful to know in terms of an individual decision making, because if you find that actually your risk according to your family is quite low, then you might do things differently, and you might make different decisions about things like having children, or it potentially it affects your decision. All right, so how did we do this personalized penetrance estimate? Well, very helpfully, a group of people, Murphy et al., had published a large combination of different cohorts of people affected by C9orf72, and that was published in 2017. And they've collected over 1,000 cases of people who were affected, mainly by ALS or FTD, and very helpfully, they had recorded all the ages of onset. And so you could put that into a graph of age here and the incidence. And interestingly, they did find that by the time you get to 80, beyond 80, you don't seem to get it. I'm not sure. I mean, that's an interesting finding, and it may or may not be absolutely true, but it seems to be that the majority of people who are going to get at least C9orf72 related conditions seem to get it in middle age, not in very late old age. And the other helpful thing about 80 years is that at the moment, that is sort of roughly the average life expectancy, at least in the UK and probably in the US. So that's quite a helpful figure to know. Beyond 80, I guess all bets are off. Something is going to get you. But it might be quite helpful to know your risk up until the age of 80, and people may find that helpful. So we thought about this, and we thought, well, actually, the family history is the most easily available information we have, and it is essentially what we do in a clinical setting already. We look at the family history and say, well, actually, your risk is probably not that high because no one else in your family has got this condition. But can we quantify that in a more helpful way? And so here's a picture of a family history that is very unrealistic, but let's start somewhere. Here's somebody who's got C9orf72, ALS and FTD at the age of 60, and we're going to assume that both their parents lived till 80, and all the grandparents lived to 80, and they weren't affected by these conditions. But we know that this expansion must have been inherited from one of the parents or the other, and they must have inherited from one of their parents. Because we don't know really of a mechanism where it could happen as a new mutation. It's got to get bigger over time, and so we assume that there must be 3 people in this picture, in this family tree, who all carry the C9orf72 expansion, but only one of them has actually been affected. And so actually it's very easy to work out the penetrance in this family up until the age of 80, because it's 1 out of 3. And so it's a third. But we can do that more formally. And we can say, well, actually, we can work out what is the chance of each of these individuals of having C9orf72. And we can work that out because we know about how genes are inherited. And we can also work out how much of their risk have they lived through, because we know their ages. And if you know that somebody has lived to age 80, then they have already lived through all of their risk, at least to age 80. And you can basically combine that information, what's the chance somebody's got the gene, plus how much of their risk have they lived through, to then work out actually what is the penetrance of the disease in this family. And don't worry about the letters at the bottom, but that's just saying that essentially the penetrance is equal to the number of affected people in the family, so it's 1 here, divided by the number of affected people plus the additional risk that has been lived through by those relatives who are at risk. And that gives you a figure for the penetrance. Now, let's just go through this. So here are the individuals who are at risk. And we know that the person who's been tested here is a carrier for the gene. But we know that this person has a 50% chance of having it. And therefore, their parents both have a 25% chance of having the same gene. And then how much of the risk has been lived through? Well, all of these people live to 80, and so they live through all of their risk. And so you can basically multiply these two things together to generate a sort of what I call the carrier survival product. And then you know also how many people are affected. It's 1, and therefore you plug them in. The penetrance is the number of affected people, 1 over the sum of the number of affected people plus the additional risk lived through by everybody else, which is all these figures joined to get added together. And lo and behold, when you do that calculation, it comes out as 1 over 3. And so when I saw this, I thought, well, actually, this is working. So I think maybe we can use this. Okay, so then just came up. These are not real families, but they could be real families, and it's slightly less straightforward than the previous example. But you might have a look at the top family and say, well, there's only 1 person in the family who's been affected, so probably the penetrance is not 100%. But in the bottom family, so lots of people have been affected, it's going to be quite high. But can we actually work it out? Well, I think we can make a good estimate by basically using the data that the Murphy et al. Group published before. So all we've done here is we've split males and females, and from that group of 1,000 or so people, and we've scaled it so that, by the age of 80 here, all the people, everyone has lived to their risk. And so what you can do is say, well, on the family history, you can see what age is somebody, how much of their risk have they lived through. So if they're 50, then they've lived to, about 25% of their risk. If they've lived to 60, they've lived through about 60 odd percent of their risk. And you can build that into the calculation. So let's just do this in this case. So here we go. Here, what's the chance of all these people having inherited the C9orf72 gene? So for the children of this person, it's 50%. For these 2 parents, it's 50%, and the siblings 50%. So that's quite straightforward. And now you use that graph. So the people who are younger, they haven't lived through very much of their risk at all, and so they hardly contribute to the calculation. But the people who are older, so this person's got to 84, he's lived through all of his risk because he's over 80. And even this person, although she died at 70, she still lived through most of her risk. So they're going to have an impact on the penetrance calculation. You multiply the carrier risk together with the risk that people have survived, and you get these figures. And you know there's only 1 person affected in the family, and so you plug that in, and this comes out as a penetrance of 45%, which I think is not unreasonable looking at it. And how about this one, where it looks like it's more penetrant? Well, again, let's plug in the risks of everyone who could have the gene, and there we go. And how about the risk that they've lived through? There we go. You multiply those 2 together, and here we've got 4 affected people. And so what is the penetrance in this? We add it all together and divide by all the risk, and this, in this setting, comes out as 77%. And I think that's not unreasonable. Now, one thing to note is that this is a snapshot, okay, because we're assuming that all these people have 50% chance or 25% chance. And of course, that information may change because somebody may have a test and they may find that they are not a carrier of C9orf72. And that information will immediately change the final calculation, because now you know something you didn't know before. And so if somebody finds that they're not a carrier, you suddenly would take away their contribution because they're not at risk. So it's just important to know this is just a sort of working snapshot of the family information. And of course, people don't know all their family information, and it can change over time. So it's very much just a working model, and you know it's not a sort of set in stone thing that will be the same forever. Okay, so the other thing you can do is basically graph this into some sort of visual picture. And if you remember those three different family trees, they all had different penetrance figures. And so all we've done here is basically is stretch or shrink the graphs of the cumulative incidence that were published in Murphy et al. Up to the given penetrance figures. And you'll see that obviously it varies quite a lot here. And in some ways it's quite helpful, because you might then be able to go and say to somebody who's 30, well, yes, your risk may be high, but actually over the next 10 years, it's not very high risk. And it's only as you get older that the risk is going to get higher. It came of I had this idea because this is what we do in cancer genetics. So in families where there are genetic mutations predisposing to breast and ovarian cancer, BRCA1, BRCA2, there's a program called CanRisk, where you can plug in all the family history and various details, and it gives you a picture of your risk over time. And that is very helpful for people to make decisions about, preventative surgery and what to do. So I'm hoping that at some point, as treatments become possible, this sort of information will kind of weigh into people's decisions as to what to do when. So I'm hoping that will be useful. And how do we conceptualize this? What does it really mean? Well, if you have the C9orf72 expansion, then you have it from birth. That's what it is. But you also have some other factors. There are going to be some other weak genetic factors that are influencing that. And there may be other strong genetic factors. Sometimes people have more than one mutation. And so that could be affecting your risk at the start. But then you have time. And over time, there may be other things that affect your risk. And we don't know what all these maybe are yet, but obviously it's an age-related condition. And so this is modifying your risk over your life. And you can either perhaps have a moderate risk over the course of your life, or you might be luckier and have a lower risk. And it's a mixture of those things you start off with, your genetic factors, and the sort of cumulative effects over your life. And you may be less lucky and have a higher risk. And I think this is what you're seeing in this variability in the families. And the difficulty will be to work out what all those things are. But this is how I am currently sort of conceptualizing this. I've said this is just a model. And so you just have to be slightly cautious about saying this is absolutely right. So although mathematically it might make sense, there is limitations. And the biggest one is probably the incompleteness of information, because so much of it depends on how much you know of your own family, and that is very variable. And as I said, it is only a snapshot. And if you were to redo the calculation in 5 years' time, it may be very different, as other people may have been diagnosed or other things may have happened. And the other thing it doesn't include is the other conditions that C9orf72 can do. So we know that some people present with more sort of Parkinson's disease type problems. And also there can be various psychiatric conditions associated. And we haven't looked into that. It's often not well recorded in these families, in the literature at least. And so we haven't dealt with that yet. But the biggest one, really, that is a problem is dementia, because dementia is just so common. And this is just showing you worldwide, what proportion of the population in certain age groups have dementia. And you'll see as you get older and older, it's really exponential, the proportion of people who have dementia. And by the time you're into your 90s, nearly most people are probably going to get dementia. So it's really hard to work out whether dementia in a family, unless it's FTD perhaps, but if it's just dementia that hasn't really been looked into, it's really hard to know if that has been caused by, for example, C9orf72 or not. So that is a difficulty. And what are we actually missing in all of this? Why is there this variability? Well, I'm hoping to be able to find out if I get any funding to do so. But you can classify it like this. So there are going to be other genetic factors. Some of those things might be actually really within the C9orf72 gene itself in terms of its sequence. The mutation may be more complex than we currently understand. And there may be effects of other genes in the sort of oligogenic, polygenic fashion. There's also going to be things that are not based on the DNA letter sequence, but are going to be epigenetic. And that means basically how turned on is a gene, how switched on is it? It's to do with how the genes are wrapped up in the cells and how they're annotated basically by the cells. And that's something it's not routinely checked in the clinical setting. It's of interest in the research setting, but we don't really understand yet how it works in terms of ALS. And then there's also things about the gene may be switched on and it may be making its product, but that can be modified by other things. So I've called this post-translational factors. This is about how a gene's product is then manipulated and processed. And those are quite hard things to measure and work out. And then there's the things that over time, might have an effect. So you're going to be exposed to various items through your diet and maybe through your occupation, whether you smoke or not. And there's all this thing about exercise, which is still quite uncertain, I think. But there are things that are going to modify things about your body and your response. And those are all things we need to do more work on. But as I said, there is this issue of what do you know about your own family history and how much has or hasn't been shared with you? And also, what about all the people with dementia or who've had other kind of problems in your family? And how do we work that out? And actually, in the literature, it's also not very clear what people mean when they say familial ALS or familial FTD. And a lot of certainly up until recently, I think a lot of those cohorts might not have included ALS or FTD, in the same setting. So if they said familial ALS, they're only looking at ALS, they weren't thinking about FTD, and vice versa. If they were looking at familial FTD, they didn't include ALS. So it's only more recently, since we've made those links, that those cohorts have perhaps come together more. But that's probably a problem for the literature, and hopefully it will get sorted out over time. All right, so I just want to finish with talking about what we're doing going forward here. And one thing we want to try to do is basically, we worked this out, we hope, for C9orf72, but we would like to try to do this for other genes. It's limited a little bit by how much information and data there is, but we should probably be able to have a good go at it for some of the other common genes. And in doing this, I also realized that in a way, we're sort of actually quantifying in a family how much what we call familiarity is there for ALS or FTD in an individual family. And actually that could be a much more useful figure to generate than to say somebody has sporadic ALS or sporadic FTD. If you could actually quantify it, then you may be able to use that information in a slightly more nuanced and helpful way when calculating this sort of thing. And then, of course, we would really like to know what is it that is actually causing the influence on the penetrance. Are there genetic factors we can find that we can actually target and use to protect, slow down or even prevent this from coming on? And if we can study the people who have the condition, who have the gene but don't develop the condition, that could be very useful. So that's what I'm hoping to do if I can get any funding to do it. But yes, I'm sure that the community must be interested in finding these things out. So that's really the end of what I had to say. I just wanted to thank the clinical colleagues I work with here in Oxford, also the people in the MND research group here, specifically Kevin Talbot, Martin Tanner, and Alex Thompson. And also I'm currently funded for my research by the National Institute of Health and Care Excellence here in Oxford, the Biomedical Research Centre. So I'll stop there, and I'm very happy to take any questions anyone's got.
Wow, thank you so much, Doctor. What a fascinating talk. We have a few in the chat. I'm sure some more will come in. I'll just start with sort of an interesting thought scenario for you. If the issue is related to something intrinsic to the mutation, which is quite possible within the C9 expansion itself, you know, there could be these people with no family history who are getting ALS or FTD kind of randomly, as would the general population, and they have no future risk, no elevated future risk, and the inheritance is limited to the people who have this other thing going on in the expansion that we don't quite understand yet. That anyway, just raise. Do you think that's possible, or do you think?
Yeah, I think that's possible. You know, we know that from other repeat expansion disorders that, for example, if you have an interruption in the middle, so you might get lots of GGGCCs in a row, but then you might get an interruption of another sequence in the middle, and that, at least in other repeat diseases, that seems to stabilize the repeat a lot. And we know, for example, in Huntington's disease, that people who have got an interruption of the CAG repeat in that condition tend not to get the condition so early. They may get it later, but it seems to be a big modifier of how that works. So I think there is research looking into whether we can see these interruptions in the C9 gene.
I will kick to Dan, Dr. Roth, who's raising his hand. So I will hit this button and you can ask your question, Mr. Dr. Roth.
I have my uncle and father and their first cousin all had the C9 or gene. I don't have, I have ALS, but I don't have the C9 mutation or any other mutation. So what's your hypothesis about why I have ALS?
Yeah, that's a really good question. And I think we are aware of families where that is the case. You think you ought to all have the same genetic mutation, but occasionally that's not the case. And I think that's showing that there are other things that are at work. And so it could be, so it sounds like several people in your family have all had the condition. And so it may be that having the C9orf72 expansion plus whatever else is present in the family, whether it's genetic or otherwise, has been enough to make the disease come out quite strongly in your family. We don't know what those other things are, and clearly it's not other obvious genes. But obviously you can also get, and most people would get ALS who haven't got a genetic mutation. And so there must be other things that are at play. And although it's unfortunate and it's unlucky, it's actually people like you would be very helpful to study, in fact. And I hope that, you know, maybe there'll be an opportunity for collecting that sort of information, because unless we have the family history and all the genetic information, we won't be able to figure it out. So yes, I think there's definitely so much more we don't know, and it's actually only with the help of the wider community we'll be able to try to piece this together.
Wonderful. There's a lot of open questions. We might not get to all of them, but we'll try to plug through until the hour. So there's a very interesting question that is your calculation based on age at death or age at symptom onset? Because you're using time. And this is a two-part question, but could you answer that part first?
Yeah, so we're looking at age of onset. We haven't looked at age of death. And interestingly, I think there is some, well, my way of thinking about it is that actually the risk of you getting something is probably different from the way that it's like to progress. So although I think for C9orf72, it tends to be quite, more often than not, it is quite a rapid progression. But I always think about it that the risk factors that actually mean that you get a condition are not necessarily the same as the things that mean it's going to progress quickly or not. And I think about this like, if you think of a flood, the things that make it rain a lot are not the same as what determines how the flood is going to affect the landscape. And so I think they're potentially two different things. And there may be things that are quite different, other genetic and other factors that are modifying progression and that are different from the risk as to whether you're going to start getting this degenerative condition.
Yeah, wonderful way of looking at it. And somebody had raised that there's the length of disease is much longer for C9FTD than C9ALS, but you said it was onset, so that takes care of that. And what do you think about repeat increasing through generations? Do you think that's likely? People were thinking that at the start, but I know that's not quite so accepted. What's your thought on it?
That's a very interesting thing because, yes, I think everybody was assuming that it ought to work the same way as other repeat expansion conditions that we know about. And I always thought that, it has a very similar sequence to things like Fragile X syndrome, lots of C's and G's. And for example, we know in Fragile X syndrome that if a male carries Fragile X and is affected by it, interestingly, whenever a male has a child, the actual expansion always decreases in size. And a male will never have an affected daughter, or at least in the daughter, it will always have shrunk. But to my knowledge, this hasn't been demonstrated in C9orf72. And likewise, this idea of anticipation, where it gets bigger, which also happens in other repeat disorders like myotonic dystrophy, I think it hasn't also been definitively shown to happen. And so my suspicion is that something is going on in the generation of sperm and eggs that is maybe resetting it to a smaller level. I have no evidence for this. The studies need to be done, but I can't see why it wouldn't follow the same pattern as every other repeat disorder unless there was something about the generation of the germ cells that is sort of resetting the size. It's still big enough that it's going to expand, but it's maybe got smaller. And it will only be if we were to actually start analyzing sperm and eggs that we could actually have a look at that.
Very, very cool. Now, there's a couple of questions to this, and I'll just open it with one theoretical here. Have you seen in the pedigrees you've looked at that there's a family with 100% penetrance, or is it always working out to some reduction from 100%?
It's hard to know because you're sort of assuming you never have all the information. So it's only the people in the more recent generations who have actually been tested. So you are assuming that the people who look to be affected are also carrying the gene. And you also always stop somewhere. People often know 2 generations back, but they usually don't know much beyond that. And so you're always missing some information. And so that will always mean it isn't 100% through this calculation because you can't make it fit. But I mean, having said that, obviously when it was first discovered, it was based on all these pedigrees where it looked like it was 100%, and that's why it was originally thought to be so high penetrance. And it's only now there's more and more testing happening that we realize it's not.
Wonderful. This goes to the point people are asking, how could they make this calculation? But it seems from what you're saying is very, very little or no family history, much lower risk, not zero most likely, and a lot of family history. It's higher, not complete, if people don't have access to like a calculator or something.
Yeah, I mean, I think you can do this yourself just looking at the family history, and this is sort of what we do. And actually, we always ask about uncles and aunts, because the more uncles and aunts you have, the kind of better news it is if they're not affected. Because we assume that, it should be a half-half of whether somebody in a certain generation is affected. And so if you have 10 uncles and aunts, even though your parent might have been affected, if they're all well, that's hopefully indicating that there's some reduced penetrance going on. And so that's it does sometimes pay to go and ask relatives about, what can you tell me about what's happened to all these others who you may otherwise not have known about. So yes, it is something that you can naturally have a look at in your own family and see. And we hope, though, we would like to be able to make this into something that could be applicable in a clinical setting. And I think the thing is that the family history is something that it's the easiest thing you can have to hand easily. It's much harder to say, oh, could you tell me someone's BMI? Can you tell me what their history of all these other the person's just not really going to have that information to hand. But usually you can give something of a family history. And so we hope this is something that would be actually useful to people having genetic counselling and give them some idea of their own risk.
One thing to think about with this, though, if there are a lot of siblings, it occurred to me when some of this first analysis that there's very low penetrance was coming out, and I looked at my own family history and many other people's where there's multiple generations impacted in a row. That could be looked at as one chance, is what I was coming up to. And like a grandparent to a parent to a child, all getting it, that's like one roll of the dice. And or could be successive rolls of dice. And you can calculate those odds. And for a low probability event in a scenario like that, it's just very unlikely. So have you thought about that in these scenarios where there's many siblings, but there's a clear line from multiple generations having it in a row?
Yes, I mean, well, I haven't formally looked at that, but I mean, it could be the case. It could be that people in that position might be very unlucky, but yes, if you are consistently finding that there are such pedigrees where it looks very strong in the actual, in the specific line that it's following, but there are lots of unaffected people when there ought to be more, that would be very interesting to see. I haven't really seen that, but we haven't gone looking for it. But if you did find that, then again, it would suggest that maybe I'm wrong, and maybe you can just have something very specific to an individual. But it's hard to know genetically how that would work, because that just sort of goes against what we understand about genetic inheritance. And so, yes, it would be very interesting if that were the case, but I haven't really seen that.
There's one item about the repeat expansion increasing over time. And is that one of the ways that you think it might be intrinsic to the mutation, about how likely it is to somatically expand or be unstable in the future?
Yeah, so yes, I think that is something that people are looking at as to whether over time it gets bigger. And again, I haven't seen definitive evidence, but it might be there. I think people have started to see that people who are younger when they're tested for this tend to have slightly smaller expansions. And so that again is suggesting that, yes, it could be that over time it's getting bigger. Again, that would fit with what we've seen in other repeat expansion disorders, and particularly in the brain. You would think, yes, it ought to get bigger and bigger over time, because of how we think it's perhaps causing DNA damage and repair, and that the repair mechanism is not maybe working properly. And that would sort of make sense. So the difficulty is that you really need to get down to the single cell level to do it, because the brain, because there's so many different cells, and you usually get this huge range So I think people are starting to look at these single cells and starting to be able to have the technology to look at the repeat in those single cells to see whether it's actually different in different cell types. And it's hard to do that in the brain over time, but you could do it in the blood and see whether, as we've only known about it for a few years. And so over time, we'll start to get repeated samples from people to see if that's actually happening.
Well, wonderful. So this has been such a fascinating discussion. I know it's late there. Can we send these other questions that have come in to you via email, and maybe we can try to get to them that way?
Sure, no problem. Yeah.
Okay, wonderful. Well, thank you so much. This is such a fascinating topic. I'm so glad very intelligent people such as yourself are working on this, and I hope you get all the funding that you're looking for. Any final word before we close?
No, just thanks for inviting me, and I hope it was useful. And yeah, hope we can work it out.